UW360 is proudly supported by Pacific Office Automation, Copy, Print, Workflow, and IT. Problem solved. BECU, a not-for-profit member-owned credit union.
They're just plastic Lego blocks, the kind children play with. But UW Assistant Professor Georg Selig says a simple toy like this can be a useful tool when trying to explain the complex world of synthetic DNA.
But what's cool about DNA is that you can actually take many of these strands and assemble them into different types of structures, sort of like you would be able to do with these type of Legos, except that everything that you do with DNA really happens at the nanoscale. So you get like these tiny, tiny structures.
Combining tiny strands of DNA to create something unique. That's what Professor Selig and a team of students are doing in the Synthetic DNA Lab, now located in the Molecular Engineering Building.
Yeah, so it actually comes in these like tiny little test tubes. So you can basically go and you can order DNA from companies, like little chunks of DNA. You get these little test tubes that have the molecule that you want in there, and then you can sort of mix these molecules up, and depending on what DNA sequence they have, they can form all sorts of interesting structures.
The challenge is finding which strands of DNA will properly combine with other strands of DNA.
If you just take random DNA and throw it together, nothing interesting is going to happen. So you have to really think about how to choose the sequences and why you choose them in a certain way to get interesting behaviors.
You can't really see any of the sequences, not even with a traditional microscope. This is nanotechnology. The size of individual atoms or molecules is typically a few nanometers long or wide, equal to 1/1000th of a meter.
But the nano controller is clearly too small to see from eyes, so we attach the DNA with some fluorescence, and then basically this machine is helping us, like it shines a laser to the fluorescence, and then this machine basically records the fluorescent signal so we can visualize in the computer. It's similar to a microscope.
The DNA strands are made up of tiny components, but they have the potential to deliver big discoveries that could one day be used in devices that could help detect cancer or other serious illnesses.
I do think that what we're doing in the longer term can give rise to new types of therapeutics and diagnostic devices, right? So they'll in some ways be helpful to hopefully to cure or diagnose disease, and I think that's obviously an important problem. You could think of a molecule, a smart molecule that you could deliver to cells, and it can distinguish between healthy cells and tumor cells, and specifically maybe kill the tumor cells but not the healthy ones.
Georg Selig says his group is about 3 years away from doing any clinical trials. The grand vision is to one day build the foundation for a technology that could be used in various ways.
We want to be able to build multi-purpose kind of devices that you can apply to various applications of interest. I mean, electronic controllers are not just good to control, I don't know, some part of your car. I mean, the whole point is that you can use that controller in many different contexts, and it will be useful over and over again, right? And that's the kind of thing that we're shooting for, like to have something, a molecular technology that can control or interact with different types of biological systems, and therefore be sort of broadly useful.